First of all, thank you for coming into this session, and let me also thank the organizers for the kind invitation to speak at this summit about our joint work on transformable 3D neuroelectronic interfaces. So just a little bit about our group. This is basically us, and below there you can see where we are. This is Munich. We're located in the south of Germany. Not so far from the Alps, although I think this image here is a little bit exaggerated. Typically, you don't see it like this. And what we're doing is we're interested in bioelectronics and sensor devices, in particular for neurotechnology and also some other applications. And just so that that we're a little bit more concrete about what kind of applications am I talking about when I talk about neurotechnology, this is just one example that we are recently have been interested in. It is the connection, the interface, the functional interface between small peripheral nerves. And in particular, we are interested in a small nerve innervating adipose tissue. So adipose tissue can be white, which is the unfortunate and normal form, but it can also become brown. And this interconversion can be triggered, or this is the hypothesis, that this can be triggered using so-called adrenergic nerves, which are innervating this tissue. So the idea is now that if we have control over this nerve, we can maybe bring this adipose tissue to transfer from white into brown, and therefore maybe help these guys here to become more like the guys on the right-hand side. And to that, we would like to establish a stimulation as well as a feedback loop so that we can control the system. So this is a little bit the general idea. So what kind of tools do we have to work in this direction? And what has been around for a very long time, since the 1970s, actually, are so-called microelectrode arrays, which can be used to couple them, for example, to cells in cell culture, but also in vivo as well. And these are some of the devices that we have been producing in our labs. Typically, you see it on silicon, but you can also do it on different materials. You can nowadays buy these arrays with thousands of channels, and you can also functionalize them with either small geometric structures or certain materials to make the interface a little bit better. Quite often, the idea is to either elicitate or record an action potential, so an electrical activity of the cell, for example, from a nerve. And what we have been thinking about for some while is, what if we don't want to put the chips now inside, but we want to investigate the cells on top of the chip? So there's several strategies. The most simple strategy is basically you build something like a multi-well plate or a single well plate on top of your microelectrode arrays, and then you get your cells, which you can get from different organs, for example, the heart or neurons, or you use induced pluripotent stem cells that are differentiated into neurons. And then you can plate them onto your microelectrode array, and you can record, for example, action potential or in general electrical activity. What you can also do is you can do it a little bit more complicated. You can build microfluidics and couple it to the microelectrode arrays. And here's just an example of how this works. This is a particular microfluidic system where we can culture one cell type, for example, in a left microfluidic chamber. And this would be here on the left-hand side. And on the other side, we could culture, for example, another type of cells. It would be, for example, interesting if we want to recreate certain pathways as they exist also in the brain, for example, from the midbrain to the striatum, where you have dopaminergic neurons, which project their axons to the other side. And the nice thing about this, if you integrate, for example, these guiding channels and you make them small enough, you can actually prohibit the individual cell bodies, these are the big green blobs that you see here on the left-hand side, to move over to the other side, and you're simply left with the axon or the neurites projecting to the other side, and thereby you can induce a little bit of polarity inside your network. Of course, this is one way to recreate very rudimentary and basic features of a network. But at the end of the day, we also want to record something. And sometimes we're not only interested in recording simply the electrical activity, but we are also interested in recording chemical signaling. And just to give you a concept about this, so if you have a synapse, for example, and the synapse is close, there are neurotransmitters that are released. For example, let's stick with the midbrain neurons. So maybe we have dopamine as a neurotransmitter. Then we would be interested in if and how often, for example, dopamine is released and we're actually getting a chemical signal out of this. So how can we record this? This is luckily very simple. Let's say this is a molecule that you would like to record. You can bias the electrode to a certain potential, which is high enough, for example, to oxidize the molecule. So once the molecule actually reaches the electrode, you can get a charge transfer, and this charge transfer can be recorded as a current. So what we do expect from this is that we get current spikes, which are dependent on the mass transport of these molecules to the microelectrode, and possibly some further kinetic limitations from the electrochemical reactions. So how does this look like? This is the examples I've shown you previously. This is our amplifier stage, and you can put all of this in. You can start to record, and what you will get is typical signals that look like this. So you have this little bit like an action potential, but it typically only has a positive side. This is due to the oxidative reaction in this case. The width of such a peak is in the millisecond range, and it is associated with the release of neurotransmitters, which are stored in these so-called synaptic vesicles when they're released. So now you can go ahead with this and, for example, probe how the dopamine release is affected if we apply a certain drug to our system. And there are several advantages of doing it this way. I've just shown you. The first one is this is a very, very simple amperometric detection. So we can use the tools that we already have at hand. It is very easy to do a lab on a chip integration to build microfluidics on top of this. And you can use it, for example, for screening drug effects. But there are also some disadvantages associated. And one is the limited selectivity as well as also the limited sensitivity. You have seen the signals were quite small. We're talking about tens of picoamps mainly, and this relates to roughly a few thousand molecules. It also restricts a little bit the device architecture if we need a single feed line for each electrode. So what you can now do is you can employ a technique called redox cycling. So here the idea is the following. You have, in addition to the one electrode that we already had previously, we design a second electrode. And we put the second electrode in the vicinity of the first electrode and put it to a negative bias potential. This means we can go into a so-called redox cycle. We oxidize on one electrode and reduce back on the other electrode. And this is kind of a recycling mechanism that amplifies our current. So since the time a molecule takes from moving from one electrode to the other depends on the distance square, if it's a diffusive process, we can actually gain quite a lot. So if we make these electrodes very, very close to each other, going into the range of tens of nanometers, we can drastically amplify the current down to the level where you can almost see single molecules. And here's just a conceptual picture of how these sensors work. So you have an oxidizing electrode at the bottom. You have a reducing electrode as the top. If the molecule comes, it can go into this recycling process and thereby be amplified. And we have built quite a lot of these systems. So either in a channel-like geometry, what you see here on the lower left-hand side is a focused ion beam cut through such an electrode structure. So you have a bottom electrode, you have a gap of roughly maybe 40 nanometers, and then you have a top electrode and the whole structure is insulated and it has an entrance. This is good if you really want to record very, very low number of molecules, but very often we are rather interested also in the temporal dynamics. And then we use different geometries, as you can see here, where you have such a porous structure where redox cycling can occur, or you can even use crossbar architectures where you can now address individual channels by just selecting individual electrodes. So this allows you to multiplex basically the signals if you're not interested in the response of each and every individual sensor spot. So the question is how to produce these things. Most of the things were produced using classical clean room fabrication techniques, either optical or electron beam lithography. But you can also print these things, which makes it interesting if you want to produce it in larger quantities, and maybe also makes it a little bit more relaxed concerning the type of materials that you can use. So if you print such an array, it basically follows always the same pattern. You start printing, first of all, your feed lines, then you print a certain electrode structure. If you want to do something complicated, then you do a spacer or a sacrificial layer and a second electrode structure. And afterwards, you insulate everything. And then you have a printed chip, which you can use, for example, for cell recordings. So these are some recordings from individual cardiomyocytes. And you see this rather regular action potential recordings in this case. So the nice thing about this is you can also do it on a lot of different materials. And quite often, we are interested in materials that are a little bit softer and a little bit more conformable than our usual silicon systems. So this is an example where such microelectrode arrays have been printed on PDMS, for example, or on so-called gummy bears. It's a candy basically made out of gelatin. So you can also directly print these things on gelatin. And you can use all of these individual microelectrode arrays for cell recordings. We typically use cardiomyocytes due to their nice and repetitive beating behavior as a test subject in these cases. And what you can, of course, also do is you can integrate these type of sensors. And I was just now focusing on the electrochemical sensors, but you can, of course, also add different types of sensors and print them directly into larger formats, such as multi-well plate formats. But everything that I've shown you so far is rather for 2D applications. So we are either probing just something where we put on our sensor, or we are probing individual cells that we plate themselves on the sensor. But it's difficult to go into deeper layers, which is often quite interesting. So we thought of using the same or similar printing techniques to extend this into the third dimension. And the way that it works is basically, for example, if you use inkjet printing, that you apply individual droplets always on top of each other, and thereby grow individual 3D electrodes. And you can, of course, it's not a parallel process, as inkjet printing per se is not very parallel unless you have very many nozzles here. But it's still possible to create rather large quantities of these pillars on such microelectrode arrays. And now you can start, for example, using intermediate spacer layers, for example, to cushion your cell, your organoid or your cell culture system, or you can even plate several cell layers on top of each other and still reach the cell layers which lie behind. And just to show you an example of how this actually looks like, this is such a microelectrode array that I've shown you previously. It's our standard geometry. And then we have printed here, this is a rather small pillar. It's just 15 micrometer in height and just a few micrometer in width. But depending on the printing technology that you use, you can make actually very, very large pillars. We have built pillars up to millimeter size, and they're surprisingly stable if you kind of do some tricks to the base here. Now, this is, first of all, quite nice for getting into maybe a little bit bigger organoids or three-dimensional cell culture systems. But we are also interested in getting this to the in vivo system. And before actually going to the mice I've shown you previously, we wanted to have a test system in which we can nicely see how well the electrodes themselves or the electrode arrays work and how good can we interface them with very small peripheral nerves. And with peripheral nerves, small peripheral nerves, I mean now nerves which are actually less than 100 micrometers. And so this is our test objects. They're very easy to breed. So you get lots of them. And they have nerves on a similar size scale that we are interested in. So here it's a little bit difficult to see. It's this whitish elongated structure that you see here on the right-hand side. This is the extensor nerve. And then you can integrate such arrays, electrode arrays, and try to couple them to the nerves. And this is now quite a challenge. Of course, there are commercial systems out there. For example, Cortec is producing peripheral nerve interfaces, all different kinds of cuff electrodes. But when you come to really, really small nerves and you also want to do recording in these systems, it's actually getting quite challenging. At least we have encountered many problems doing this. So we said, okay, the good thing is now we can 3D print all the geometries, or more or less all the geometries that we would like. So we investigated a little bit, what are the best cuffs that we can 3D print for inserting these very small nerves, which are below 100 micrometer. And we have played around quite a lot with different geometries and tested all of them out and found a winner in the end, where we have such a scenario. This is basically the cuff. The entrance here, this is where the nerve is actually inserted, is now significantly less than the size of the nerve. And we have integrated the 3D electrodes into the base of the substrate. This is typically a PN foil of 50 micrometers. So now we have a cuff electrode and you can couple it. And the nice thing about these desert locusts is now that you can actually have a biomarker where you can check how well you can stimulate from the nerve. You can also do the recording from the nerve, but it's a little bit more difficult to interpret what the signals, what the action potentials that you record are actually doing. On the stimulation side, this is very nice because basically the position of the leg is frequency coded. So if we apply a very simple feedback control loop, we can modify or modulate the pulse frequency with which we stimulate the nerve. We can see how the locus reacts. And basically you can do this via a set point where you measure with a flexible resistor the extension of the leg, feed the signal back, doing error control and amplification, and then look at the actual movement of the individual leg. And this is very repeatable. You can do this for a very long time. And this allows you to see how well your interface actually works when you're starting to change the geometry. And while this is nice, it has one disadvantage. And the disadvantage is that the cuffs that I've previously shown you, once they're implanted, it is very difficult to remove them, at least to remove them without damaging the nerve. And for that, we went back and thought a little bit about a different design. And in this design, we again 3D printed a cuff electrode, but now we printed additionally on the backside a super swelling material. And we did that in such a geometry that the cuff electrode, once you start to immerse it into the water or the electrolyte of the insect, it basically starts curling up. So you have a few tens of seconds and then the cuff builds itself. And I would like to show you a quick video of how this works. So here you see such an implementation procedure. And you see here are the electrodes, and they're now processed with the super absorbing material. And now once it comes into contact, you have a couple, or let's say 10 of seconds, then it actually starts to wrap around by itself. And now we have, in this case, it's again a little bit difficult to see, but maybe you can see it here. We have this very small extensor nerve captured in our probe. And the good thing is now, because we can induce a lot of flexibility, this is a very stable connection, and you can do all the operations that I've shown you before. But now what you can also do, if you want to remove it, you just pull it. Of course, this elicits some nerve response, but in principle, the material is so flexible that even these very small peripheral nerves are not damaged, and you can actually pull it out again. And you can close up the locus, and they're surprisingly sturdy. They can live like this, and they still can move their legs. Okay, where is this going? We hope we can in the future use these transformable and long-term stable neuroelectronic interfaces in the system that I've shown you previously for the adrenergic nerve. Would be nice to include it, of course, with further biomarkers and a feedback control. And by this, we hope we can go into the intervention and try to trigger interconversion, for example, from white to brown adipose tissue. So with this, I'm at the end. I would like to thank again the people who did most of the work. This is a little bit older picture, so they're not too many faces on here which are actually contributed to the newer part of this work, but Leroy Grob and Francisco Zurita, they were working on the 3D electrodes that I've shown you previously. We had a lot of support also by external collaborators, by funding agencies. And of course, we are also collaborating on this project very strongly with Tetsuhiko Teshima and NTT Research in general. And with this, I'm at the end. And I'd like to thank you for your attention, and I'm open for questions.
Sorry, if anyone in the audience has questions, I can bring you a microphone so that everyone can hear. Any questions from the audience? Okay. Oh, yeah.
So this is Kit Parker from Harvard. Can you tell me a little bit about how you're controlling the biotic-abiotic interface on your chips? And I guess what I'm looking for is what kind of extracellular matrix proteins you're putting down on there. Thank you.
Yeah. So yeah, this is a very good question. So it depends very much on the system we're talking about. So for these particular locus in vivo system, we don't do anything. We don't do any coatings. But for other systems, depends, for example, on the cardiac systems I've shown you in the beginning, we typically have coatings like fibronectin, or we do just some protein coatings. Sometimes we have PEG coatings. That depends a little bit on what the cells like and what causes problems. For the locus themselves, we don't need to worry too much about it. They're super sturdy. You can leave it implanted for at least the length of the experiment, so several weeks, and you will not see any aversive side effects.